Welcome to the inaugural Robert M. Storm Distinguished Lecture Series. We think this will be an annual event because of the generous gift by Bill Lovejoy, who's back there. Bill, raise your hand there. Bill was one of Doc's 71 graduate students, and I thought I had a lot. We recognize Doc Storm because of his contributions to the Department of Zoology, where he began in 1939 and was active for 6 decades. So he's recognized probably as the foremost authority on vertebrate biology in the Pacific Northwest, mostly for herpetology, but he's worked with all the vertebrates. And we have lots of his former students in the audience. This is really great. We have family members right here. And this should be a fun situation here. On a personal note, I was a young whippersnapper professor when I first got here, and Doc was active. And I had worked with mammals all my life, and I was getting sick of getting bitten and sick, and I couldn't catch them. So I asked 2 people to help me switch my research program. One was Doc Storm, and one was Rick O'Hara, who's over there. Rick. He was one of Storm's students, one of his later students. He also worked with John Weens, and they taught me everything they could. I didn't learn everything, but they taught me all about amphibians, or I would be probably not tenured and a mammologist somewhere. But Rick's been at Chemeketa for a while, and he was really instrumental in me doing a better job than I did when I first got here, and Doc knew where you can get the animals and the natural history of all these animals. And on another personal note, before I stop this thing, I also performed the wedding ceremony of Patty and Doc in the year 2000. So we were friends, we were colleagues, and I'd like to introduce Bill Lovejoy now so he can say a few words.
Thank you, Andy. A tribute to Doc Storm. Some of you may not know how Storm came to be called Doc. He earned that title not for his degree, but as a term of endearment and respect for his service as a medic on the beaches of Normandy during World War II. From hence on, he was known as Doc. Of the many professors I've been privileged to study under in both geology and biology, 2 of them stand out above all others. One was a paleontologist at the University of New Mexico. The other is Doc Storm. Both these men presented a broad interest in subject matter, but also inspired and motivated students to pursue their interest in the field and in the laboratory as well. Doc had a rapport with students, both in and out of the classroom, that I tried to emulate in my teaching career at Georgia Southern. I consider myself fortunate to have shared campfires with Doc on many of his field trips and on hunting trips as well. I remember during one pheasant hunt, I managed to get in Doc's line of fire at a rising rooster and was pelted with shot from his 12 gauge. Doc and I had our differences, but I thought that was carrying them a bit too far. However, both the pheasant and I escaped without serious injury. Doc wasn't one to dispense advice to his students, with one notable exception. He advised those of us planning to make a career of teaching to always check our fly before entering the classroom. Good advice for a young professor, and I followed it religiously. But, excuse me, lost my place. As I neared retirement, I was more inclined to agree with what Winston Churchill said when an aide reminded him that his fly was unzipped. Churchill replied, "Dead birds don't fall out of their nests." By establishing distinguished lecture series at these two universities, New Mexico and Oregon State, it is my fervent wish and expectation that such inspiration will continue to motivate students for years to come. I think it all begins with Dr. Butch Brody. During my visits with Doc in January of this year, I was reminded of a poem by Walter Savage Landor, written on the event of his 75th birthday. I strove with none, for none was worth my strife. Nature I loved, next to nature, art. I warm both hands before the fire of life. It sinks, and I'm ready to depart. No one who knew Doc Storm can doubt that he loved nature, nor can they doubt that he warmed his hands before the fire of life. If anyone lived life to the fullest, it was Doc Storm. However, I'm not so certain he is ready to depart. On the contrary, he's fighting long and hard, right up to the end. I suspect the Grim Reaper will have to drag him kicking and screaming all the way to those happy hunting grounds. Good hunting, Doc.
Well, thank you, Bill, for that those very moving remarks. And so it is my pleasure. I'm Bob Mason, a member of Integrative Biology here. It's my pleasure to introduce this afternoon's inaugural speaker in the Doc Storm seminar series. I would just like to say for both, Bill is obviously was a student of Doc's, and now Butch Brody was a former student of Doc's, and we welcome them back as their alma mater. So it's our special privilege to welcome the inaugural speaker, Edmund D. Butch Brody, to deliver the inaugural address, and so the inaugural seminar. And as you've heard, both Bill and Butch were students of Doc's. Lots of faces in the audience here knew and love Doc very much. So it's a special honor for us to have Butch come back. But in reflecting on his career, Butch really is a great representative for this first seminar series in our newly formed Department of Integrative Biology. So a lot of you people think you're in the zoology department, but you're actually in our new Integrative Biology department. And so Butch's research, you'll see, truly is integrative in the spirit with which we formed this new department. So you're going to see everything from molecular biology all the way up to population and evolutionary biology and things in between. So he is a perfect representative of what our department stands for. So to get the other things out of the way, so Butch is a very accomplished researcher. He's published well over 200 papers, very well known for, he's actually like Doc too. I was surprised in looking through his CV how many other things he'd done. I certainly knew him and his son, another Butch Brody, for their garter snake newt interaction work, which I'm not going to say more about because he's going to tell us all about it today. They've been funded for many years, for several decades now, by NSF. Very interesting work. And let's see. So I would say, I think at that I'm going to allow Butch to actually talk to us about interesting things and not these introductory remarks. So Butch Brody from Utah State University is here to deliver his lecture, and I don't have the title up here. I've seen the posters all around, so you guys have seen the titles too, so I'm just going to hand it over to, there we go, yeah. Arms Races and the Evolution of Tetrodotoxin Resistance. So Butch, welcome.
Thank you.
Actually, I think you don't need this.
So I think I'm adequately mic'd up. Wave if I am. Okay, we're mic'd up for some reason. I'm sure my voice will project I've always been a loudmouth, and you'll be able to get me in the back. So, arms races and the evolution of tetrodotoxin resistance. You know, that title could have been a dozen different things, kind of like that. The important thing here is that my son co-authors all my talks because much of this I simply couldn't have done without him. He was actually born a year after I started this research, and I had to wait until he got out of graduate school to collaborate with me so we could really expand this research. So we start at the beginning, since in a real sense my beginning is here. Here's a story one of my grad students found on some media site on true interesting things. And of course, it's the story that I've always told when asked, why did you start looking at newts? And basically, this is an old wives' tale related to me when I was an undergraduate by Dr. Walker, who somehow convinced Doc Storm that he should accept this football player from Monmouth as a graduate student. I won't relate what my grades were in college, but suffice to say today, they wouldn't even look at my application. Doc took a chance on me and said that I should continue looking at whether or not newts are poisonous for a master's thesis. And these two gentlemen got me started in such a way that I'm still looking for the answers. That says more about me than them, probably. So tetrodotoxin is the absolute key to everything I'll be talking about today. It is the compound found in the skin of newts that garter snakes have evolved a resistance to allow them to eat the newts. So the interface of coevolution is tetrodotoxin. And most of this I'll give you in another slide. Okay, tetrodotoxin occurs in all these types of organisms that have red on them, from bacteria to dinoflagellates to some red algae, various animals across here, including salamanders, our friend Ptericha granulosa, it's found in some frogs, and we've got a different color dashed line here and here because animals in those groups have evolved resistance to tetrodotoxin in their prey. So this gives you an idea as to the extent of variation. Now, I'm not a neurobiologist, I'll say very little about this, but tetrodotoxin binds in the sodium channels and has the effect of paralyzing the organism. Resistance to tetrodotoxin requires that amino acids in the pore region of the sodium channel, so these are 4 domains that twist around and form one pore where these come together. And by altering or by having altered amino acids, it prevents the tetrodotoxin from binding and blocking the pore. So that's for the back. For me, first, it was all about the newts. Are they poisonous? Well, we learned very quickly that they're poisonous. The first time I ground up newt skin in a mortar and pestle and took a glass syringe and injected a mouse, which died in my hands before I could put it on the table. You know, that will get the hair on the back of your neck to stand up a little when that happens. So we learned that pretty quickly. And then we learned that most newts around the world are poisonous. So those in orange are called the modern newts. I didn't name them, somebody named them. They're called the modern newts, trust me. They all have tetrodotoxin. So if we look back at least 69, maybe to 90 million years ago, all newts had tetrodotoxin. All the lineages that have split past that have tetrodotoxin. We'll come back to that point at the very end of my talk because it's important. But also important is newts cannot have tetrodotoxin unless they're resistant to tetrodotoxin. And these changes in the sodium channel compared to the salamander ambystoma are the ones that have taken place, and they took place very stepwise, and you can trace which base pair changed from one step to another, from Salamandra to the Plurideles Echinotriton group onto the modern newts. It's possible to trace those changes. So this is the background that we're looking at. So what are we doing now? We're with the newts, we're looking at histological sections. This is a granular gland, and it turns out that the granular glands are where most of the tetrodotoxin is found in a newt. Now, I'm carefully avoiding saying that newts produce tetrodotoxin. If you ask me my opinion, I would say they do, but I'm not giving you opinions here. So this is where they're found. This is a scanning EM shot of a freeze fracture showing the granules up in that area of the granular gland. I just love that picture. I had him, I mean, they're black and white, but I had it colored in OSU orange for you. Or nude orange, as it would be. So, with collaborations with a group of colleagues in Brazil, they have worked out the technique to stain the tissues for tetrodotoxin, and they use it with an immunohistological technique that they've explained to me, but I can't explain to you. But they now have the technique worked out where we will be able to tell, is this tetrodotoxin or is it found in the matrix? And that's big for me to understand, because then we're going to be able to look at other amphibians that have tetrodotoxin and look at the distribution there. We're also in that attempt to learn, do newts produce their own tetrodotoxin? We're looking at transcriptomes between populations of newts that have tetrodotoxin and those that don't have tetrodotoxin, in the hopes that we'll be able to determine where in the genome there's DNA that predicts presence or absence of tetrodotoxin. So we know newts have tetrodotoxin. Obviously, I wouldn't be giving a seminar if snakes didn't eat newts. So when we look at the coevolution, let's look first at the prey, and we expect to see in a coevolution system geographic variation in toxicity. We expect to see resistance. I've already shown you a slide on that. There are multiple newt species involved. That would be predicted. And the prey are dangerous to the predators. And for a predator-prey coevolution arms race to evolve, we believe that prey must be dangerous to the predator. There must be some cost to the predator. Okay. So, with John Thompson's geographic mosaic theory, there should be coevolutionary hot spots and cold spots. That means there should be evolutionary relationships taking place at some localities where selection is acting on newts and on snakes, and at other localities we don't expect to see that. We should have polymorphism. If there isn't variation, there likely isn't evolution. There should be populations at different stages of the evolution of the relationship. So let's see if we see that. So we first looked at newts, at toxicity, measured the tetrodotoxin in a newt. And we found a range from, sorry, we can't measure that, out here to, this is like 6 or 8 milligrams. The lethal dose for a human is 1/2 milligram. So there are populations of newts that average in the 6 or 7 milligram range. That population is found in Soap Creek ponds out by Camp Adair. So when Doc Storm and Doc Walker started me on this path, I was really fortunate to be going to school here and not in Washington or California, because had I tried to do this work there, this would have been a really short seminar. But by starting here and being very lucky, man, this is where newts are hot. So, but you can see that across their range, there's enormous variation. And you see here areas that are more brightly colored have more toxic newts. So basically, the Willamette Valley and the south coast of Oregon, and a little bit down here south of San Francisco, and then we get a lot of areas with very low toxicity. So a real strange range and distribution. We've now actually tested newts clear up into southern Alaska, and it's also very low there, as you would expect. So we know the situation in newts. We'll come back to it, but let's now look at the predator. The predator that we've studied most is Thamnophis sertalis, which you see here. Common here around this area. We again would expect to see a geographic variation in resistance. We would expect the resistance to be by amino acid substitution in the sodium channel. We have looked at multiple species of garter snakes, but first I'll start with sertalis. So these are all the same species from different areas along the west coast. Thamnophis sertalis in all of these areas eat newts. Their populations vary by resistance. We know that there's a cost to resistant, although I won't show you the data today, because I heard the dean was a statistician and I don't want him to review my stats. Sorry. But it turns out that if you take a group of baby garter snakes, some of them crawl faster than other ones. The slowest ones have the highest resistance to tetrodotoxin. So there is a cost in crawl speed related to resistance. Now, we measure resistance with this very fancy racetrack, the snake racetrack. It has electronic sensors along it, so I just have to chase a snake up the racetrack and the computer tells me how fast they crawled. I'm actually chasing a baby snake there. Then we give it a small dose of tetrodotoxin and measure how that dose slowed the snake down. And I can slow a snake down whether it's very resistant or whether it's not resistant at all, as long as I'm careful to not start by giving them too much. If you give them too much, then you only are able to test them in one run because they die. But if you use very small doses, They recover within 24 hours and can be tested again. So this is the beauty with tetrodotoxin, is there's full recovery from small doses. So, and by doing this, we're able to measure individual differences between snakes. So we're not just looking at population averages, we're looking at individual differences. So if we look at how resistant garter snakes are, one would be equivalent to the amount of toxin it would take to kill a mouse. It's actually a mass-adjusted mouse unit because snakes weigh different amounts, often less than a mouse, often more. So this is mass-adjusted. So all snakes are a little bit more resistant than mice, but you've got to come over here before you're resistant enough to really eat a newt. And then you see up at the top some very, very resistant animals. I'll talk about what that means momentarily. So we've got snake resistance, we've got newt toxicity. We were really happy when we printed these two off. You know, look at that. They're great. They reflect each other. And then we decided to go a step farther and say, so do these really match? Are these levels of resistance and toxicity really a match? And we were a little bit surprised when we did that because we found that areas that were very high in resistance weren't really a match. They were exaggerations. So we talk about hotspots of coevolution. That's where the selective pressure is the greatest. So the selection on the newt is greater, the selection on the snake is greater, and this takes place where the levels are matched. So it takes place right here, and it takes place here, the South Coast, here, the Willamette Valley, and up around the mouth of the Columbia. Most of the areas where snakes and newts interact, there's no selection going on at all. They're far too different in their levels. If we analyze the data farther, and so you see this zone here, that's where we would predict that there was some selection going on. Selection for greater toxicity, selection for greater resistance. Below this line, any snake in the population would be unaffected by any newt there. Above the line, newts would be lethal to all the snakes in the population. Now, note that there are no populations above the line. There are a lot of populations below the line. Basically, the newt never wins. The snake always wins, or they're still locked in this battle. So we have here a group that's essentially lower. There's no interaction, but these populations here around the San Francisco Bay Area are really curious because the newts are pretty toxic here, or this way, but the snakes are really resistant. So what we think happened, and is still happening, is we move from ancestral conditions where the newts increased the level of TTX, got into this zone of reciprocal selection, moved along, and then there was a 1 amino acid change in these snakes, and it changed their level of resistance so much that they escaped the arms race. There is no newt in the world that will affect these snakes. In fact, a large adult female here could eat the amount of tetrodotoxin that would kill 900 people. So there is no newt anywhere near that. 50 people, yeah, we could get that from a newt. 900, no. So they have simply evolved, escaped the arms race, and happily crawl around eating any newt they see. But there's also, from these snakes, no selection on the newt. Now, we'll give you a little more detail in a few minutes. So, I've got a lot of stuff I want to jump, so some of my transitions could leave something to be desired. So we have looked not only at Thamnophis sertalis, the common garter snake, but we've also looked at other garter snakes here in the West that are sympatric with newts. So the blue line and the green are newt distributions. Sertalis is resistant. Thamnophis atratus, that's atratus, is resistant. Thamnophis couchi is resistant. And just last summer I was able to test some Hammondi from down here in Southern California, and they're also resistant. So a number of different species have evolved resistance independently and differently by changing different amino acids in the sodium channel. When we consider these various species that have evolved resistance and are now interacting with the newts, we have attempted to see if they're applying different levels of selection. So this is a bit involved. I'm going to simplify it in a minute, but we're looking at 4 different species up here: Sertalis couchi, atratus elegans, and we're looking at different levels. Don't worry about the symbols, but note where we get various colors. In Northern California, all of the garter snakes there, and this fits the newts in the area, the garter snakes have low levels of resistance. The newts have a fairly low level of toxicity, but there's some real coevolution going up on here because it's still matching in a small area right here. In the Sierras, all of the snakes in the southern Sierras are highly resistant. And this goes so far as to, let me point that pyramid out. That's elegans in the Sierra. They're highly resistant to tetrodotoxin. Nowhere else in their range. As far as I know, they don't eat newts. So we really don't know what's happening there. But it's very curious. But the most interesting is in what is going on in this coastal California region, where some of the populations are very high in resistance, but they're all variable. So regardless of what species of garter snake we're looking at, some individuals have very high resistance, some individuals have very low resistance. So, of course, we looked at it. If we contrasted Atratus in green and Sertalis in red, we find that they have the same range of resistance from about 3 or 4 mass-adjusted mouse units to nearly 1,000 mass-adjusted mouse units. The newts, if we plotted the newts in this, they would fall right here. So 75% of the Atratus have a low resistance. They can't eat newts, but these can eat newts. We find both of those in Sertalis, but the vast majority of Sertalis have higher resistance and are able to eat newts. So are these species at different stages in their coevolution with newts? Did these super resistant genes recently evolve? Have they been there? Are they just in a polymorphism? We don't really know. But there's a dissertation or two understanding that. So this is my favorite individual garter snake ever. You saw him on the posters. Her photo has been in both Science and Nature, and that's more than any of us can say for ourselves. So why are some garter snakes so brightly colored? Well, naturally, I think it's because they eat a lot of newts and are themselves poisonous. So here's a field-caught animal that we caught that had a level of TTX in the liver more than twice the lethal dose for a hawk. The snakes retain and sequester tetrodotoxin in their livers. Do they have a mechanism? These are things we don't know. Do they have a mechanism for moving and storing the TTX. We know that pufferfish do. No one has looked at newts or at snakes. Is this situation only in Sertalis, or is it found in Atratus and Couchi? And does it occur in all the populations or only in populations that are very resistant? So there are a ton of questions left. Animals other than newts also have tetrodotoxin. So here's another newt from Japan, a frog from Southeast Asia, another tiny little frog from Brazil, another frog from Central America. These all have tetrodotoxin in their skin, the same compound as in the newt. And for each of these where we know there's a resistant predator, snake, that's feeding on them, and these snakes all have evolved amino acid replacements in their sodium channel. Now, different ones, but they've all become resistant by a similar mechanism. If we look at the distribution of those changes on this model of a sodium channel, and I'm confident that models exactly what a sodium channel looks like, but you note that all of the changes are in very isolated areas, nothing down here. All of the changes that confer resistance in snakes are apparently constrained to this region. That we don't understand. And it's odd because in pufferfish, there are changes down in this region that confer resistance to them. So up to this point in my talk, we've only looked at sodium channels in the muscle tissues. We didn't have the technique to look at nerve channels when we started doing this. So we just looked at the muscle, assuming that if a muscle was resistant, a nerve had to be. When we finally got around and got the technique worked out, primarily in my son's lab, none in my lab, I might add. So, and we looked at NAV 1.7, 1.6, and then the skeletal muscle, and we looked across snakes. We didn't look at every species of snake, but we looked at a ton of hundreds of snakes. And what we discovered is in the small peripheral nerve, so this cartoon up here shows a rendition of a small peripheral nerve. All reptiles have NAV 1.7 channels that are resistant to tetrodotoxin. And that includes this very famous reptile on the top line, the chicken. So about, 200 million years ago? From that point forward, reptiles have had peripheral nerves that are resistant to tetrodotoxin. Trust me, the newt had nothing to do with that. This, for reasons I don't know. mammals do not have resistant channels in 1.7. So 1.6, which are the myelinated nerves, evolved resistance in those lineages that tend to eat amphibians about 38 million years ago. And we're showing you those in green. And then the sodium channels in the skeletal muscles are found only in those species that are feeding on amphibians with tetrodotoxin. But the stage was set for the evolution of this change by the preceding changes in the 1.6 and 1.7 channels. So now we understand it all. We understand that these snakes are resistant. In order to feed on tetrodotoxic prey, they need resistant sodium channels at all those levels, except for these. This we really don't understand. So hognose snakes, in areas where they're sympatric with newts, feed on newts, and are tremendously resistant, but they have no changes in their muscle sodium channels. Their sodium channels cannot be resistant to tetrodotoxin. There is some other factor going on that we're trying to determine, but this we discovered quite recently. It's not the only case of this. There's another case, but again, more work to do. So we've at this point, we've looked at all of these relationships. What we haven't looked at is, are other predators important? Now, there are other things in garter snakes that eat newts. I mean, that's an adult male newt, the great blue heron. Those are otters eating newts. There's chomping a newt, chomping a newt. That's actually not a newt, but it's enjoying it anyway. So what is it about those? Well, it turns out the places where birds and mammals can eat newts, the newts all fall in this range. Now, I'm not going out to taste one, but they all fall in that range. So other predators, in fact, one of you today asked me, are the larvae of newts toxic? And I said, well, yeah, kinda. So they have low levels of toxicity, much more right as they hatch from the egg, and then it's reduced, and then it stays fairly constant at a low level. So we did some trials to see if it was possible that a predator, dragonfly naiad here, would distinguish between different levels of toxicity in these larval newts. And if you look at it, they ate these, they didn't eat these. So after the trial, we retrieved the newts and measured the amount of tetrodotoxin in them. So, and these were all same population. They were lab reared. So there could very definitely be some selection going on just within the larvae. It's probably what's going on in the eggs is probably even more important. There's high variation between clutches, and there's a correlation between female toxicity on the x-axis and egg toxicity on the y. So you see for each female, her eggs are relatively similar, but it varies from female to female. So what? Well, it turns out there are caddisfly larvae that eat Tarica eggs. They eat a lot of Tarica eggs. The wild-caught caddisfly larvae have tetrodotoxin, so we go out here to Soap Creek Ponds near Adair. We pick up caddisfly larvae. We check them. They have tetrodotoxin. In the lab, they can eat newt eggs. All it does is increases their growth rate. There's geographic variation. Caddisflies from here can eat newt eggs. Caddisflies from Utah die when they try and eat newt eggs. And there's geographic variation in the sodium channels, just like we see in garter snakes. So, is this a big deal that a few caddisflies are eating newt eggs? So, this is out where we've worked, just north of town here. My former student looking for caddisfly larvae and newts and whatever. There are published records on these ponds that nearly 5,000 newts were taken at one time from one pond. There are a ton of newts out there. I'm not sure there's still that many, but I can remember in grad school there were a ton of newts. So that's a lot of newts. 5,000, but there are more caddisflies. We did population estimates on caddisflies, and we're what, 700,000 to 800,000 caddisflies? It turns out, if you do all of the calculations, the newts could produce around 1 million eggs per year. And the caddisflies in one of those ponds could eat 1 million eggs in 36 hours. So resistant caddisflies is a really big deal for newts. Caddisflies not only eat newt eggs, but once they're full, they attach a couple to their cases and carry them around with them till they're hungry. Now, this happened in the lab. We've never seen it in nature, but they attach these, and then a couple days later, they crawl out and eat them. So I like that picture. So we had one of these things with the snakes. We've also got it with the caddisfly. So we've looked at all of these relationships with caddisflies that's really a talk in itself. But the important thing is that caddisflies are resistant where they're with very toxic newts. So is there coevolution going on between the caddis and the newts? There's variation in number of eggs consumed. They're resistant sodium channels only in sympatric populations. Those caddis that eat more eggs grow larger, presumably would have more offspring. What I don't have up here is they do select lower toxicity eggs if you give them a choice between highly toxic and moderately toxic. They can sense the amount of tetrodotoxin in the egg. Now, newts have egg toxicity linked to adult toxicity. We haven't sorted out any genetics, but we know there's a survival difference between high and low toxicity. Newts also deposit their eggs higher in the water column on vegetation when there are caddis or caddis odor in the water. This allows them to escape the caddis because caddis don't climb all that well. So, do we now then have coevolution where newts are in the middle and we got things eating them from both ends, where the caddis are out here eating and favoring the highest toxicity eggs, which were deposited by the highest toxicity females, and presumably will give rise to more toxic females. And then you've got garter snakes eating newts. The lowest toxicity newts are the ones eaten. Those at the very top tend to be avoided. So we get all of these various types of selection going on, all based around tetrodotoxin molecules. So are garter snakes resistant to tetrodotoxin because caddisflies were resistant first? So caddisflies have been around for 225 million years. Newts, maybe 100. Garter snakes, maybe 25. Garter snakes are not responsible for newts being toxic, or maybe even have elevated toxicity. So I think we need to continue to study this because it's really interesting, but we need to understand the evolution, we might need to turn our attention back a little earlier. So a ton of people have worked on this and more are working on it. And I want to say one more thing about Doc Storm. I think this is something that he was most proud of and took pleasure in. And this is a few years old. I may have missed something, but this is a distribution of where his students and their students are working. And I only looked at academic assignments here. So there are profs in all these states that are related. Okay, I'll turn it back over to you. And
All right, questions for Butch?
yeah.
Well, considering PTX is present in many different organisms, are the genes that are responsible to make the compounds?
They're not known. No, I wish they were. Yeah, we just don't know. So tetrodotoxin has been synthesized in a lab under high heat and pressure conditions, but the biological synthesis of it is not known to my understanding.
So if you don't know what genes produce the tetrodotoxin, could it be also something in the newt's diet, or could something in the newt's diet influence the amount of tetrodotoxin that's making, if it is?
Yeah, so someone is going to think that I planted that question, but I didn't. So the Japanese investigators are pretty convinced that the presence of bacteria accounts for the level of toxin in pufferfish. So I did a calculation, and if you filled Gill Coliseum down there with a bacterial culture at the highest level that bacteria can produce, it would not be enough to supply 1 newt. So I'm thinking that's not going on in those ponds. Whether they're getting a food source is a different question, and we have certainly looked at it. We've kept newts for years in my lab and fed them worms that have no tetrodotoxin because we checked them, and they continue to increase their level, the newts. Are there precursors? I don't know how to get at that one yet, but it's the part of this we do not understand.
If you crossbreed the non-resistant caddisfly larvae with the resistant, obviously to the adults, does it reduce their resistance?
Yeah, they're different species. Caddisflies are more of a problem than snakes. Snakes have to. Now, let me say that we have crossed Thamnophis sertalis, garter snakes, from different areas, and what we got was offspring that were more resistant than either parent.
So it's a dominant gene, maybe?
Yeah, or a series of them, I think. Yeah.
So you've given me quite a scare. I take my granddaughter out to catch newts all the time. Obviously, you've caught a lot of newts.
Caught a bunch.
So at what point does this newt become a danger to humans?
So there have been 3 people die from ingesting newts. Alcohol was involved in all 3 cases.
You love those students too, weren't you?
Oh, I hope so. No, they weren't OSU students. A couple of them took place down near where I grew up in Myrtle Point, and you know, a couple drunk fishermen out on the Rogue River said, "Oh, I'll bet you can't eat that." "Oh, yeah, I could eat that." Well, yeah, he could eat it. So seriously, I wash my hands after I handle a newt before I eat something. If I have cuts on my hands, I try not to handle newts. I have cut myself badly with a razor blade that I was cutting newts. It's wonderful. There's no pain whatsoever because it deadens that nerve, just bang. The problem is you can't get the bleeding stopped because it expands the capillaries. So, you know, with your granddaughter, wash your hands. Newts taste terrible. She's not going to eat it. You know, it'll be another drunk fisherman before we lose another person. Yeah.
But 2 questions. Do those brightly colored garter snakes have a bad taste? And the other one is,
You know, as you may remember, because this is one of my old professors, who I didn't recognize until I heard him speak, and then I recognized him immediately. As you may remember, I hate snakes. So I wouldn't get a snake anywhere near my mouth. But I suspect they probably do taste bad. Yeah.
Then the other question is, do you think those guys really did die from drinking coffee that had a newt in the water?
We don't know. I don't know. Doc Walker told me that 50-some years ago. And, you know, it doesn't matter if it was true or not. It caused me to start working. So I'm okay with that.
Right, the different kinds of snakes in the same regions where 70% or so are highly resistant and 25 to 30% are less, and otherwise 75, 35, right? Might they have different kinds of diets in terms of some eat a lot of newts and some eat a lot of other things, or is there any way to think about this other ways?
So we've actually done that at that site, and both of them eat a lot of newts. The ones that are less resistant are primarily eating the larvae of newts, and the highly resistant ones are eating the adults. The other thing that goes on with those species of garter snakes there in California is you don't find them together in the same pond. You'll find one species or the other. And the larger species is very aggressive to the smaller one and will drive them away from in a chamber. They'll drive them to the other end of the test chamber. So there's a lot ecologically going on. We've just scratched the surface on that one.
Is there a similar escape from the evolutionary arms race in caddisflies as there was with the garter snakes? In that, are there caddisfly species that are completely resistant to any amount of tetrodotoxin?
You know, we don't know yet. So it's been fairly recent when we discovered that. And we haven't even published the sequence data. There are a couple other twists going on there. But California requires permits to collect caddisfly larvae. Not only that, you have to tell them what species you're going to collect and where before you get the permit. And no one, well, not no one, but very few people can tell one caddisfly from another. If we were going down to fish and we ripped one up and threw it out, that's fine. But yeah, so we've run into some issues.
So I'm interested in that evolutionary, breaking out of that arms race too. And you mentioned that there was a single amino acid that was responsible for that in those populations. So I was curious what the nature of that was, where that was relative to the other evolution.
So I can tell you the paper to look at.
Okay. Well, and my second part of that question was, does it appear to be convergent?
Is what?
Does it appear to be convergent? What's the evolutionary relatedness between those populations relative to the larger geographic region?
They are, so the two species that have evolved super resistance do it differently. I think that's what you're asking me. They certainly have converged phenotypically. So they have the same trait caused by changes in the same molecule, the same stretch of DNA, but the changes were different.
What about toxicity in fish from eating a caddisfly?
Fish we're eating.
I don't know. I don't know. I assume that a caddis pattern will still take a trout in where there are toxic newts. What I do know is that if you take a caddisfly larvae into the lab, you feed it a newt egg, you let it develop and eclose, when you get an adult caddisfly, it still has tetrodotoxin. So it's being retained. You know, could it protect them at some level, or their eggs? I don't know. I don't know when this thing's gonna stop. You know, it's like a escalating thing going on, and I keep promising my wife I am gonna retire. You know, I did, not tomorrow, but I am gonna retire when we get all the answers.
One more.
You're one of the few who's done this experiment. I remember reading once you took Northwest salamanders, this microcilia, ground it up, put it on your tongue. I think you published that. I've heard rumors how you reacted, but have you ever tried it with a newt? I've always wondered, like a dog or something, if it grabbed the animal, touched it to its tongue. Have you done that, or?
No.
I just wondered if there's an immediate
there's one
reaction
report
to your tongue, because it doesn't affect your hand.
No, there's a report in 1890. Omerud is the woman's name, Miss Omerud, at the zoo in London. She took one of the European newts, a Triturus at the time, held it between her teeth until it secreted, and then had a really bad reaction to it. And I had read that, and before, and you're right, I've tasted an animal or two, but not newts, because that first day when I injected a mouse and got ready to set it down to see how long it would live, and it was already dead, the chance of me tasting a newt after that was nil.
Let's thank Butch for a really nice
Thank you.
Thanks, Butch.